So you know those sci-fi movies where they have, cloned humans growing in vats so that they can harvest organs from them, and it's all pretty scary and dystopian. Well, for a long time I thought, well, we're a long way from doing that sort of stuff. I mean, how could we possibly do that? We don't know how to grow body parts outside of the body in vats, and and we still don't. But we might not be that far away from being able to do this sort of thing, to grow human organs outside the body. What we would do is grow them in farm animals. So you could grow human organs inside pigs. And I've come across some research that was, there's a video of a seminar of a guy that's interested in doing this sort of stuff and has done a lot of work progressing this idea. It was on the internet, on YouTube, just early 2021. Yeah, so this is a reminder about why people are interested in doing this sort of work, because of the massive shortage of donor organs. So in the USA, you can see, this graph shows, the growing waiting list for human organs. So, imagine if you're in this pink zone here, you're waiting for an organ, and in fact in the USA, 20 people a day die waiting for an organ. So, I mean, this research may sound all scary and a bit of a yuck factor, but just consider how you would feel about the yuck factor if you're on this list, if you're in the pink zone, or you've got a kid in the pink zone. And also remind you, even if everyone that was capable of donating an organ were on the donation list, probably still wouldn't be able to solve the demand problem, because only 3 in 1,000 people die in a fashion that their organs are suitable for donation. So I mean, this is a serious problem, and people do sell their organs. There's a whole black market in organs. So yeah, I mean, it is an issue that would be worth solving. So people need to put themselves in other people's shoes a bit when they're in the abstract. Oh, this is all yucky and we shouldn't be doing this, sort of playing God. Well, medicine has played God for a long time. So this is the work of a Dr. Nakauchi. Actually a lie, it's not actually the work of Dr. Nakauchi, it's a work of him and all these people that work in his lab, and as science gets done by lots of people. But this group that actually managed as a proof of concept to grow a mouse pancreas inside a rat, and then take that mouse pancreas out and transplant bits of it back into a diabetic mouse, and then cure that mouse of diabetes, and in a scenario where they don't need to supply anti-rejection drugs to the mouse. So this is kind of like the holy grail of what this sort of research would be. You could grow, if this would pan out into humans, you would grow a human pancreas inside a pig, and then you transplant that human pancreas back into people and cure them of their diabetes. Now, this is all based on the ability to create what are called chimeras. Now, of course, chimera is a monster from mythology, but in biology we have a specific meaning of what a chimera is, and it's an animal derived from a population of cells that derive from more than one species. So it's basically a mixture of cells in the animal. So this makes it different to a hybrid, because a hybrid, you cross two animals together, like you might cross a lion and a tiger, and you end up with a liger. Each one of the cells in your liger is half tiger and half lion, where if you had a chimera between a lion and a tiger, some of the cells would be lion cells and some of the cells would be tiger cells. So that is an important difference. A chimera is a mixture of cells, not cells that are a mixture of genes. How do we create chimeras? Well, I'll let the doctor explain, because he explains it pretty well.
So I just tell you how we make chimeras. For example, if we want to make mouse to rat chimeras, we prepare mouse pluripotent stem cells. It could be iPS cells or ES cells.
So what he's talking about here is these stem cells that everyone's really excited about. These are pluripotent stem cells. They're stem cells that can give rise to any organ in the body. And so he's talking about those. You can have mouse versions of those. And he's talking about could be the iPS cells. So they're the induced pluripotent stem cells. They're ones that can be derived, as a clone of a person. You can take a skin cell, and you can make a stem cell from those skin cells. So you can induce them to go back to stem cells. Or there could be those ES cells, the embryonic stem cells, ones that are derived from fetuses.
We also prepare early embryos from rats. We call it blast stage embryo, which is about 3 to 4 days after conception. So this rat blastocyst has a small cavity. This is still a cluster of maybe 100 cells, and this portion becomes the body, and the other part will become a placenta. So anyway, we inject mouse iPS cells into the cavity of this blastocyst, and then this is how we do it. This is the rat blastocyst, and we are injecting mouse pluripotent stem cells through this tiny, pipette. You can see these cells being injected. And then we transfer this chimeric embryo to a foster mother. 3 weeks later, we see birth of chimeras, and many of them grow into adult, and they look like this. It's a mouse to rat chimeras.
So yeah, that's all well and good and cool and all, and you've got a rat or a mouse that's got some of the cells are derived from the other creature, but that is not too much use to you, because if you look at the pancreas in one of these chimeras, some of the pancreas would be derived from the mouse and some from the rat, and we want 100% mouse pancreas inside the rat, or you want 100% human pancreas inside your pig. So we need to figure out a way to ensure that you get 100% of the donor organ in your recipient, and this is where this great idea came along, and it's based on the fact that people have done basic research to figure out the genetics of organ development to some degree. So we've got these mutants of genes that are responsible for the first stage of organ development. So there's a mutation in a gene that's responsible for kicking off development of the pancreas, and this gene was actually first discovered in Xenopus laevis, which is a clawed frog. So that's a pretty obscure place to first discover this. But we know this gene. If you don't have this gene in the mouse, then no pancreas develops. So you can have these lines of mice that will die soon after birth because they've got no pancreas. But if you start off with that line of mouse, and you get a embryo from that line of mouse, a blastocyst from that line of mouse, and you inject it with the rat cells and make a chimera between the rat and the mouse, then the rat cells actually develop where the pancreas would go, and you get 100% rat pancreas. Or if you do the reverse, you get 100% mouse pancreas inside the rat. So this is like totally cool. So basically you got this organ deficient line of animal, and then you can rescue it with the organs from a different creature, and that's what they went ahead and did to develop this work further. So yeah, they went ahead and did this experiment, and were able to produce a mouse pancreas inside a rat, and then able to take that mouse pancreas out and transplant it back into diabetic mice. And then they're able to see whether the diabetic mice could actually control their blood glucose levels. And this is a graph for this result. There's a bunch of different animals, I think, on this graph, so it all looks a little bit complicated. But you can basically see that this is the days after transplantation of the pancreas that was grown in the rat, and you can see their blood glucose levels go way down, so they are controlling their blood glucose levels. And at these two time points here, they actually went and operated on the mice and removed the graft they put in there. They removed the bits of pancreas they put in there, and their blood glucose levels spiked up again. So this is just proof to themselves that the transplant they'd made was responsible for the controlling of the glucose. So yeah, a pretty impressive proof of concept of this sort of technology. So yeah, this work was done back in 2017, and so you'd ask where to from here. Well, of course, you obviously want to do this sort of work in larger animals than mice. So the idea that these scientists had was that you'd do this in livestock animals. So you'd maybe get pigs or sheep to grow these human organs inside them. And so people have moved forward to try and test this out in pigs. This came across my radar briefly at one point, where people saying, "Oh, look, human-pig hybrids" was in the news, and I kind of dismissed it as completely crackpot ideas, because I know you can't make a human-pig hybrid where you cross a human and a pig, but it turned out that the press were sort of misusing the name hybrid when they really, as I said, they really should be talking about human-pig chimeras. So yeah, there've been some press on this sort of large animal version of it, the human pig work. So there was this paper, this newspaper article in the Daily Mail that is actually from early 2021. But there's also been this article back in 2016 from The Guardian talking about this work. And this work is from a guy by the name of Pablo J. Ross. He's from the UC Davis in California. He's actually worked with Dr. Nakauchi, done some collaboration with him on this large animal version of it. And and they've been working on these on trying to do the chimeras between humans and these large animals. They've obviously been working on this for quite a while, trying to do it in pigs. And it's actually quite disappointing, actually. It looks like they're having trouble getting the sort of level of chimerism in the human pig or human sheep mixture. And I guess it's maybe not that surprising. I mean, the initial work was done with rats and mice, and they're quite closely related on the evolutionary scale. And because this all really relies on the fact that, a rat pancreas can grow inside a mouse. So all the things that signal to a rat to grow a pancreas are also more or less going to work inside a mouse, which, it's kind of surprising. I mean, it's kind of understandable that it might work, but on the other hand, it's kind of surprising that it does work. So, I mean, it's all basically down to the fact of evolution. We know that, rats and mice are closely related, but when we shift to getting human cells to grow inside pigs, it doesn't seem to work quite as well. The number of human cells that end up inside the pig chimera are very, very low. So this seems to be a bit of a barrier. And I don't know where they're going to take it from here. The people on this paper discuss the possibility, the reasons why this might be the case, the possible differences. I don't know whether they're planning on seeing if they could maybe modify the pigs so they're less, so they're a bit more human-like in terms of being able to support the human organs growing in them. But also, I note from the research published here and talked about here is that basically to sort of confirm the idea, it's all about evolutionary distance. People have been looking at chimeras between chimpanzees and other non-human primates and find that, if you pick primates that are not too distantly related, you can get a higher level of chimerism. I don't think anyone's done the human to chimpanzee chimera yet, or the human to macaque chimera. But that is kind of the obvious place to take this. And this is kind of a bit of a bait and switch relative to what we were promised at the beginning, wasn't it? We were promised at the beginning it's growth of human organs in livestock. And I don't know how I feel about that bait and switch. Maybe I'm being a bit specious, because I get a bit more squirmish when I think about, maybe I get my replacement organ grown in a chimpanzee, because we're not eating millions of chimpanzees every day, whereas if we're growing it in a pig, we are killing millions of pigs to eat. But maybe that's just a irky gut reaction, because pigs and chimps are both alive. So I don't know. Let me know how you feel about the ethics of that. No one's actually said they wanted to going to do this in grow them in chimps. Not in any of the research I've read, but kind of that's the implication of where this has ended up right now. That perhaps not surprising, it's a little bit harder to grow a human organ in a pig than it is to grow a mouse organ in a rat, or a rat organ in a mouse. Let me know what you think in the comments.